Now we will begin Session 5, Mitochondria and Healthy Longevity. I'd like to introduce our session chair, Dr. Abe Takaaki from Tohoku University, who will be guiding us through this session. Our speaker for this session is Dr. Vadim Gladyshev from Brigham and Women's Hospital, Harvard Medical School. Dr. Abe, over to you.
Thank you very much, Dr. Mishima and Dr. Hirano. Today, we'd like to have our program on the healthy longevity through restoring mitochondrial function and longevity. In Japan, where the population is aging, it is urgently needed to increase the healthy longevity and also narrow the gap between the average life expectancy and healthy life expectancy. Aging causes several physical damages like sarcopenia, hearing loss, cancer, atherosclerosis, and infection. And its improvement is expected to reduce care requiring period, improving QOL, and reducing social welfare budget. In this scenario, we believe that a common predisposing factor may be a decline in the mitochondrial function. Mitochondrial abnormality has been reported in the so-called mitochondrial diseases, but not only mitochondrial diseases, but also in many kinds of diseases, hearing loss, diabetes, kidney diseases like CKD, cancer, neuromuscular diseases. Therefore, drugs, diet, and rehabilitation that restore mitochondrial function are the central dogma of healthy life expectancy with maintaining QOL, quality of life. To date, however, there is a very tiny, virtually no treatment for the mitochondrial disease that was approved by FDA or covered by the health insurance. As therapy agent for the mitochondrial disease, we prescribe, for example, CoQ10, that's an electron donor and substrate for the mitochondrial electron transfer system, or idibinom, that's an CoQ10 analogs. And recently, EPI 743, that is expected to act on NQO1, and also it stimulates the glutathione synthesis that may reduce the mitochondrial loss. However, there has been no specific and effective drugs. Recently, this peptide drugs SS31 that is bound to the cardiolipin, that is a lipid very common to the mitochondrial cristae. And that drugs is very effective for the models of ischemic reperfusion or diabetic diseases by changing mitochondrial structure changed. Simultaneously, we also have developed a drug termed MA5, which also binds to the mitophilin, that's the protein in the cristae. MA5 binds to the mitophilin structurally and enhance the electron gradient and help the ATP production. In addition, MA5 also accelerates the dimer formation of the ATP synthesis. These morphological changes supported the biosynthesis of ATP and ameliorate the symptoms of mitochondrial deficiencies. So far, MA5 shows the cytoprotective and organ protective effects such as heart, renal, cardiac, and liver diseases, as well as hearing and ALS and some other diseases. In addition, viral infection hijack mitochondria and manipulate the replication and change the redistribution, and caused by decreasing the membrane potential and increase loss, and also the cellular damage through NLRP3 inflammasome activation and production of inflammatory cytokines. Dr. Yasuda, our collaborator, conducted the SARS-CoV-2 infection experiment and found that our compounds are cell protecting effect with same as molnupiravir, and also it's effective to BA1 and BA2 mutant. Thus, improving mitochondrial function may be also useful for the infection disease and also treating as their sequelae. So the development of this mitochondrial homing agent AMED is fully supported by AMED, and now we are now going the first in human phase 1 clinical trial, and it's currently underway, and that will be finished. I hope finished within this year. These will suggest that AMED may be a new mitochondrial homing drug that improves the mitochondrial function and may be a new therapeutic agent for many symptoms caused by aging. Accordingly, in my AMED Moonshot project, blood samples, urine samples, breath, stool, and any other human viral samples will be collected from 400 cancer patients, 150 dementia, 150 IBS people, and we integrated data of the mitochondria and also gut microbiome linkage. That is very important for the longevity experiment. So I would like to examine who will be effective of my drugs or not. This makes it possible also identify the disease markers and drug treat target. Simultaneously, we would like to mention about aging and how to measure it. Aging differs among individuals and organs, and some humans have a good aging and others are not, even at the same chronological age. In the past decades, many people doing the genomic, proteomics, and metabolomic method to find tons of thousands of markers for aging. Among the aging markers, recently, Dr. Horvath et al., also his Vadim, also proposed the use of DNA methylation clock to predict the age. So far, using this age is very conservative with all the species, and also he will show that the calorie restriction is also stop the maturation age specific to each organ. Using this approach, maturation in blood is associated with cancer, cardiovascular disease, all-cause mortality, and this is supporting that this age acceleration is strongly linked to the common diseases. So that's why we'd like to combine with our samples analysis and mitochondrial analysis, mitomics, and also his techniques to measure how predict the lifespan and life expectancy and mortality. So this AMED have launched this Moonshot program, and we do conduct research and social implementation to extend health and life expectancy without frailty by 2040. Okay, I'll finish my talk and transfer my talk to Dr. Vadim. Dr. Vadim, please.
Thank you. Good afternoon. It's really an honor to be here and participate in this amazing conference, and of course to visit Japan as well. So thank you very much. So it's very clear that many people in this audience, they work on cancer or various chronic diseases or various other conditions. Interestingly, these conditions and diseases have a single major risk factor, which is often overlooked. Of course, this factor is aging. So aging is by far, it dwarfs anything else, any other risk factors. So if we try to understand aging and try to target it, we could potentially delay the onset of all of these diseases at once. Of course, then we need to ask, but what is aging? There is no consensus in the field currently. So intuitively, we understand that it's kind of simple. We see this every day. But in fact, if you ask experts in the field to define this, it's very difficult. So these are the common definitions that people utilize. And you can see that they are widely different. Some people think that aging is the increased chance of dying. Some other people think of age-related changes, or maybe loss of function. Some people think it's a continuation of development or accumulation of damage, and so on. Of course, these features, they're all related to one another, but there must be one kind of major process. And this is one of the goals of my lab, to understand this and to be able to target it. So this is the way we kind of think in the lab about the aging process. We kind of think that it all comes from molecular kind of damage in a broader sense. This is basically a negative consequence of being alive. Things accumulate as byproducts, so as mutations, epimutations, and gene expression changes, and so on. We call it the deleteriome, combination of deleterious changes. And they unavoidably accumulate over time. And they result eventually in functional decline, and then diseases, and then death. But of course, if we are able to target it, we need to think about this from the molecular perspective, from this kind of damage perspective. And so we developed a research program to address that question. So the way that we work is that we work to understand longevity. And we do this at the level of different cell types. For example, some cells in the blood, they live short life, but cells like in the brain, like neurons, they can live the whole life. Longevity across mammals. Some mammals have lifespan of only 2 years, and some 200 years. So nature is able to modulate lifespan very, very dramatically, 100-fold. The question, how does it do it? And then we also try to understand longevity across interventions, because we already know many different ways, genetic, pharmacological, dietary, lifestyle, that extend lifespan. So we can understand this at that level. So this results in what we call signatures of longevity. These are molecular patterns, typically omics patterns, for example, at the level of the epigenome, or metabolome, and transcriptome, which indicate the potential to live long. Yeah? It's not age-related changes. This is something that we could modify and make the organism live longer eventually. At the same time, we also study aging, and these are actually age-related changes. And we do this by studying and developing epigenetic clock, as Dr. Abe already mentioned, also transcriptomic clocks, and also causal biomarkers of aging. This results in actionable biomarkers of aging that then we utilize, for example, to screen compounds and monitor how organisms, tissues, and cells age. And the third component is rejuvenation. This is really a new topic in the field, where we not only can understand progression from being young to old, but also potentially understand progression from being older to younger, typically not for the whole organisms, but maybe for a subset of the organisms. And I will give you a few examples of that. And this allows us to target aging and rejuvenation and identify dietary, pharmacological, and genetic tools and features that we use in this area. So the way we kind of started this approach, we started looking at exceptionally long-lived mammals. We sequenced over the years many genomes and published many papers on the topic. And so, for example, this was the first paper we published over 10 years ago on the naked mole rat. This is the exceptionally long-lived mammal, the size of a mouse. It lives more than 40 years. There's another one called Damaraland mole rat. This is the bowhead whale, which lives more than 200 years. This is another one called Brand's bat. It's one of the tiniest mammals, weighs only 5 grams, lives more than 40 years. So what kind of from this conclusion from these studies is that different mammals appear to accumulate unique features that allow them to live long. This is not necessarily the same features. But at the same time, we also find kind of coordinated changes across species. And this is our most recent study we just published last month, where we have done a comparison across 41 species of mammals. And we've done it at the level of 3 organs, brain, liver, and kidney. These are omics patterns, primarily the transcriptome, where we identified gene expression signatures that kind of nature utilizes to extend lifespan. And these are some of the genes shown here. For example, this is RPL28 gene that increases with increase in lifespan across different organs, brain, liver, and kidney, some other genes. But anyway, so we have like hundreds of genes like that. Another approach is to look at interventions that are already known to extend lifespan. And this study is a comparative analysis of the interventions in mice. These are 17 ways that are known to extend lifespan. And this is an aggregated correlation, Spearman correlation matrix. And you can see that many interventions, they form cluster. So they kind of modify the mouse in the same way or similar way. These are things like calorie restriction or methionine restriction, treatment with acarbose or canagliflozin, or FGF21 overexpression, and things like this. So they modify mouse similarly, but then there are other interventions which also extend lifespan, but apparently do this at the molecular level differently. Things like rapamycin treatment, or maybe protoenzyme, or CMYC deficiency. They also associated with longevity, but mechanisms are different. Like across mammals, we also see that there are many ways to longevity, also across interventions. Now, we try to combine all of these features, and here, it's a complicated slide, but it's a very important slide. So here, we look at the longevity across species. You see, like at the level of the brain residual, kidney residual, liver, and so on. Of course, they related to one another. This means that the changes that we observe, they are conserved across tissues and across species. Next one, we look at the aging patterns, and we look at the aging in the muscle across mammals, brain across mammals, across human tissues, different tissues, global across tissues and species, and so on, muscle, mouse, liver, kidney, anyway. So they all correlate to one another, also form cluster. And the third cluster is the so-called longevity signatures. These are signatures like growth hormone deficiency, calorie restriction, median lifespan, and so on. They also correlate to one another, typically, except for rapamycin here. So what we observe, interestingly, is that not only they form 3 clusters, but, for example, longevity across species positively correlates with aging. So the longer-lived species are literally older based on omics, not younger. So this is a big surprise in the field. The third conclusion is that longevity across species does not correlate with longevity across interventions. What we find in the lab in terms of simple interventions, like a simple knockout or overexpression or treatment with exercise, sleep, whatever, is different from what nature does in extending lifespan so dramatically. So this whole potential of what nature does is essentially untapped currently. And we are trying to utilize these approaches in the lab. Also, this is a kind of the broad view of longevity and aging and molecular pathways together and how they related to one another. For example, this is maximal lifespan, aging signature. This is interventions, longevity interventions. And you can see some pathways that appear as dominant across of these comparisons. Things like, for example, mitochondrial translation, going back to Dr. Abe's presentation, oxidative phosphorylation, they're intimately linked to maximal lifespan interventions. But also we see other features like, for example, DNA repair becomes important, interferon gamma, amino acid metabolism, and so on. So why is it important? Why do we need even look at these longevity signatures? Actually, these signatures allow to directly identify interventions that extend lifespan. So because it's omics patterns, we can screen, for example, a public database and ask a question, like which manipulations are consistent and similar to the signatures that we observe. And for example, we screen like public databases and find hypoxia. Mild hypoxia is very similar to many longevity interventions. Has never been tested for lifespan extension, but this would be an excellent candidate, for example. Or we could screen, we've done a chemical screening for thousands of compounds, identified some interventions like this. We just published last month, for example, that this is from a paper a few years ago, but we just found that actually this predicted compound does extend lifespan. And we are testing additional compounds. Actually, we have now a large project on that. So, but if we identify interventions, we need other tools to check in advance. Not waiting until like mice die or other animals die. In humans, it's impossible. Of course, humans live too long. We want quickly to test whether interventions change biological age of cells or tissues or organisms. And for this, we need what we call biomarkers of aging or clocks. The concept of the epigenetic clock has been pioneered by Steve Horvath. Used to be at UCLA, now at Altos Labs. But we've been working at the mouse level primarily. And this is the example of our first epigenetic clock. So here, we analyze 16 mouse groups, and you can see the actual age corresponds to the predicted age really well. And these 4 groups are mice that have been subjected to calorie restriction. So at any age, they are biologically younger. We know that calorie restriction extends lifespan, but we don't need to wait until mice die. We could test them at 10 months old. We already know that the mice are younger, literally younger. And this is a way to test it. And for example, here, this is a chronologically matched mice, but this is a wild-type mouse. This is growth hormone receptor knockout mouse. This mouse is 40% longer lived, and at the age of, relatively young age, they are biologically also younger than control mice. Again, so we could easily see this. Here, we use the same clock to test the conversion of fibroblasts to induced pluripotent stem cells. We basically used Yamanaka factors to convert these cells from the aged mouse fibroblasts from lung or kidney, and you can see that the predicted age of iPS cells is 0, as it should be. So the clocks, even though developed for other tissues, also work in iPS cells. More recently, we developed a clock that works at the single cell level. So, and it quantifies DNA methylation. This is a paper published about a year and a half ago. The tool is called SCH, for single cell age clock. So what we find based on this clock is that actually amazing heterogeneity across tissues, across cells in the tissue. Many cells age like this, but there are some cells, like damaged cells, they seem to age faster. And there are some cells, for example, many stem cells, that age slower, like muscle stem cells, for example, apparently they age slower. So it's a huge, when we look at the tissue, it's like this. It's a huge heterogeneity in aging. There is another study I want to highlight, just published a couple of months ago, where we find that biological age, not like chronological age that steadily increases, actually fluctuates. Under conditions of severe stress, organisms literally become older, and once the stress is relieved, the biological age goes down. And we describe 4 examples here, like severe surgery, COVID-19 infection, not just infection, but when somebody goes like ICU and ventilator, the biological age is increased, and then once the person recovers, actually decrease. Also pregnancy. Apparently women in the 3rd trimester are biologically older, become older a little bit, and postpartum the age decreases also. And the 4th model that we describe is parabiosis, when we connect young and old mice, so they establish common circulation, and in this way, younger mouse becomes kind of older, and then once we separate, it can recover, and then kind of the age goes back to normal. So, and speaking of parabiosis, we also have another study in Nature Aging, which actually will be published next week, where we study the effect of younger mouse on older mouse. Here we connect young and old mice for 3 months so that they develop common circulation for 3 months. And after 3 months, we separate them and follow for the rest of their lives. What we find is that these mice, older mice, after they have been having common circulation with older mice, they live longer. Their lifespan is extended. And also, they are literally younger based on the epigenetic clocks that we developed. For example, we look at the liver. This is at the stage when they were 3 months together. And this is when 2 months later, the effect still persists. So they literally become younger and then continue to be younger and live longer. So rejuvenation is possible here. So this is another example of rejuvenation that we just recently discovered. We found that at the stage of zygote, which is a fertilized egg, the predicted biological age is very low, but it's not 0. And then during initial stages of embryogenesis, it kind of stays the same up to like blastocyst, and then after implantation it goes down to the minimal biological age. We call it ground 0, around the stage of gastrulation. And then what we think is aging actually begins at this point. This is another big question in the field. When does aging begin? There is no agreement. But I think we found at least that the lowest biological age corresponds to approximately the stage of gastrulation. We published a few papers on this in mice, in human, and also currently kind of working in frogs. But again, more data still needed to validate this concept, but we're very interested in this topic. I will give you just a couple of examples on that. Here we utilize our single-cell epigenetic clock to test this idea. So we look at the epiblast, 4.5 days, 5.5, 6.5, 7.5. You see there is rejuvenation. Cells become younger during this period. When we look at all three germ layers, they are also rejuvenated. But when we look at the extraembryonic tissues, they are not. It seems like this rejuvenation during embryogenesis is also cell type specific. Also, we recently developed, this is still unpublished, a very good transcriptomic clock. And currently, most of the studies that we do, we combine epigenetic clock and transcriptomic clock. These are kind of orthogonal methods to predict the biological age of tissues. And you can see the clock is also very, very good. This is based on more than 3,000 samples where it's adjusted for lifespan based on the interventions that extend lifespan. So it's very good transcriptomic clock. And this is just one application of the clock to the embryonic rejuvenation, as I mentioned. You can see we also, like in the case of epigenetic clock, we also observe that during early embryogenesis, there is rejuvenation up to the ground zero, and then aging begins. So it will be very interesting to understand the nature of this rejuvenation process, or exactly what actually happens. And we are very interested in that because eventually, if we understand, maybe we could apply this to somatic cells like in the adult state. Now I'd like to highlight another question, which is, I think, very, very critical in the aging field. And this is the question of causality. So here is the mid-journey image of the same person in the younger and older state. Let's compare them. For example, we can see that the hair becomes gray, and there are also loss of hair. Yeah? But is this causal? It's probably not, because it doesn't really affect the aging of a person. We see other changes. For example, the wearing of glasses, hearing aid, or we could add this, for example, like a walking cane or wheelchair, or maybe number of visits to doctors. So these are age-related changes, and if we build clocks, they end up at the molecular level in the clock, but they are not causal. For example, if we fire all doctors, there will be no glasses, no hearing aid, no walking cane or wheelchair, and based on this clock, everybody will be young, but it's not. So what is important is that we cannot just follow age-related changes. We need to understand the nature of these changes. Because changes could be damaging, changes can be neutral, or changes could be adaptive, protective changes. Yeah? And for this, we developed a first causal clock, epigenetic causal clock that quantifies biological age in a causal way. Here it was done by Albertine, who is a talented graduate student. The paper is still under review, but you can see an early version in the bioarchive, where he combined Mendelian randomization genetic approaches, these approaches that I just already introduced to you in terms of the epigenetic clock, to identify causal epigenetic changes. And these are features that, on one hand, may reduce lifespan based on accumulation of damage, and on the other hand, protect against it. And the clock based on the damaging changes, we call it damage here, and adaptive changes called adaptage. So you can see that if we use these CPG sites, whether they are damaging or adaptive, we could perfectly predict the age of a person. However, if we apply these clocks in the rejuvenation mode, like here, we again apply Yamanaka factors to convert fibroblasts to iPS cells. The damage clock shows rejuvenation, but adapt age does not. So again, it just highlights that we cannot just simply utilize age-related changes. We really need to understand the nature of these changes in a causal way. I highlighted to you one problem in the field in terms of the clocks, in terms of understanding how the clocks work. Because to me, that's a revolution in the field. For the first time, we are able to quantify the aging process and check longevity interventions. But we don't understand exactly what we are measuring. And these are some of the challenges which we need as a field to solve. This is a very active area of research currently. I don't have time to discuss them, maybe during Q&A session, but anyway, so I just wanted to highlight one more point here. We developed this tool called ClockBase. And here we basically apply all the major clocks that have been published to the entire GEO database. The paper is still unpublished, but you can go to this website And if you're interested in anything, literally in the GEO database, any data set, you can go and look at the predicted biological ages of the system, whether this is a treatment or cancer, diabetes, whatever is in that database. So that's it. So this is my lab. What I told you, the work of Albert Ying, Jesse Paganik, Alex Tishkovsky, and Bohan Zeng, primarily, and other people work on other topics, as well as the previous lab members, these people contributed. Of course, we're also grateful to collaborators, Dr. Takaaki Abe, and also Tomoka Kasahara from Tohoku, but Steve Horvath, and many other excellent collaborators, and also funding sources. Thank you very much for your attention.
Thank you for the presentation. We'll now proceed to the Q&A session. Before we begin with the audience questions, we have questions from Dr. Abe to Dr. Gladyshev. Dr. Abe, please go ahead with your question.
Okay, thank you very much, Dr. Vadim. So I think we have many questions from the floor, but initially I'd like to ask some question for you. Initially, in iteration clock, there is now many series or many different clocks. So made by person. Do you have any idea to combine one unified clock or not, especially in human, because it's hard to understand?
Yeah, it's actually an excellent question and a very critical kind of issue in the field. You're right. So there are several clocks. For example, in human, there is the most famous clocks like a Grimm age or Pheno age, Dunedin pace, the Horvath clock, and so on. We also developed a few clocks as I presented to you. So which one is better? Which one is to use for in testing interventions? There is no consensus in the field. And I don't have an answer because the answer would be biased. So one way to address that, we recently formed a consortium of leading labs that work on clocks. So about 25 labs, and kind of to bring them and discuss the main issues like what is biological age, what is a biomarker of aging, what is predictive prognostic biomarkers, and so what are the tools that allow us to validate these clocks in terms of in clinical settings. So we have a first paper also almost accepted in Cell on the topic, and the subsequent kind of studies continue. I mean, in the end, there's no good answer. It's an ongoing process currently, but it's a very critical issue because, we need the consensus and we need to find what are the kind of best biomarkers that should be utilized.
Okay. I'd like to ask one more question. I'd like to ask about the period of the administration. In rat or mouse, I think 3 months or 6 months is necessary to change the epigenetic clock in your experiment. However, in your data, in the human COVID data, it's just only 2 weeks or 3 weeks. Only 2 weeks or 3 weeks change the clock in human. What's the difference between the human experiment and the mouse experiment?
Another good question. It really depends on the experiment. So, for example, in many interventions, in some interventions in mice, like for example, calorie restriction, we test. We don't see an effect after 1 month. If we treat for 6 months, we see an effect. So, if the treatment is kind of very, very strong, then the effect may be more observable. Like, for example, in the case of, like discussed pregnancy or like a hip fracture, or I don't know, somebody goes on ventilator being sick in COVID. So those have very strong effect, and they can be observed more rapidly. The weaker effects, like for example, many interventions take longer time, to observe.
Okay, thank you very much.